Absolute pleasure to introduce Longqi Liu. Dr. Longqi is the director of the Institute of Cell Science and the group leader of the single cell genomics at BGI Research. Dr. Liu obtained his PhD from the Chinese Academy of Science, where he investigated the roles of epigenetic enzymes in somatic cell reprogramming. He then did his postdoctoral training at the BGI Shenzhen, where he studied the dynamics of epigenome and transcriptome during human pre-implantation embryo development. His research focuses on the development of technologies for high throughput single-cell multi-omics and its applications in the study of cell fate complexity. And Dr. Liu will present on COVID-19 related cell atlas research in maca fasciculari. So Dr. Liu, please.
Thank you, Kathy. So can you see my screen? So first, I would like to share.
Not yet, but I think you will. Yeah.
Okay, now can you see my screen?
Yeah, wonderful.
Okay, great. Thanks for Katie for the nice introduction, and thanks for the organizers for inviting me to this wonderful virtual meeting. So my talk today is single cell atlas of a non-human primate. So sequencing technology is probably the most important milestone during the last few decades. And thanks to the efforts from scientists all over the world, we have now completed the genomic sequence for many species, particularly the model organisms. And this data resource has greatly improved our understanding of life complexity, such as evolution and gene function. As we know, cells are the functional unit of life. And if we look into the difference across species in the cellular level, you will find a tremendous difference regarding cell number and cell types. And this is very interesting, but still we have very big challenge to answer many questions. For example, how can we define those cell types, and what's the molecular mechanism underlying those cell identity? So this question is very important for understanding of cell fate complexity, and I think the basis of human diseases. So with the rapid development of a high throughput single cell sequencing technologies and the reduction of sequencing cost, now it has become possible for us to really provide the cell atlas of a whole organism. Actually, previous efforts have achieved the single cell atlas of many species, such as lower animals, for example, C. elegans, planary, Drosophila, or zebrafish. And this has been help us to understand, the cellular complexity, for example, the lineage differentiation into embryos, of embryos. And actually, in the past few years, people have also begun to map the cell atlas for mammalian species. For example, the human cell atlas is aiming to, generate a fine map of human cells at single cell and spatial resolution. Also, to get a global overview of the cell types, actually some groups have have completed the cell atlas of mammals. For example, Guoji's lab have generated the cell atlas, organ cell atlas for many species, for some species such as mouse and human. However, so far the cell atlas of non-human primate is still lacking. So we know that the non-human primates, such as macaque, actually are very close to humans, and because it can break through some, ethnic restrictions, so it becomes a very important model for the human-related studies. And previously, people have used the monk model to study, for example, the disease-responsive genes, or do preclinical drug screening, and this has helped us to understand, disease complexity and development of new therapy methods for many diseases. And we and our collaborators have mapped the single cell multi-omics for macaque early embryo development, and I think this is important data research to study human embryo development. And we have also generated the single-cell chromatin accessibility and transcriptional atlas for macaque cortex. And with this data, we can find the relevant cell types for many neurological disease. And also, we are quite interested in the complexity of organs in macaque. So we are trying to generate the whole, the atlas of all the organs in this organism. But actually, because we know that there's a huge number of cells in monkey, so so high throughput single cell sequencing technology and a cost-effective single cell system is really important for us to really provide the huge cell number. So for this reason, we developed DMB Lab C4, which is a negative pressure driven droplet system. And this system can help profiling thousands of cells at a time. And now it has been, it can be used for the profiling of single cell RNA-seq, single cell ATAC-seq, and single cell multiomics, including gene expression and chromatin accessibility. And also combined with our self-developed sequencer, DMB 61 system. This is ultra high throughput single cell sequencer. And we built a single cell data generation center in China National Gene Bank. And this help us to do large scale single cell data generation every day. And based on our single cell sequencing platform, we performed single cell or single nucleus RNA-seq for the monkey, macaque, fascicularis. And we provide 40 organs from 8 major systems. And we eventually get a total number of nuclei or cells of over 1 million, which means that for each organ, we have over 20,000 cells. And we did clustering and for the single cells, and we identified over 70 cell types. And in a global overview of the data set, you can see that each organ clustered separately, but with the exception of many common cell types, including macrophages and cellular cells, stromal cells. And actually, this is, I think for each organ, we also have tens of thousands of cells. And for example, in liver, we got over 90,000 of cells. And I think this number is quite big, and it really enables us to precise identification of cell type within these organs. We actually identified quite similar cell composition pattern in these organs, and this is quite consistent with previous published cell atlas. And we also covered some organ cell atlas that are not available in human, for example, the carotid pituitary gland. And I think this atlas is also important supplement to human cell atlas. And as mentioned, there are a lot of common cell types across the tissue. Actually, these cell types share many overlapping marked genes, but still they show quite big tissue specificity. For example, the endothelial cells, we can see the endothelial cells exist in organs such as lung, liver, kidney, but actually they show quite tissue-specific markers, probably because the microenvironment of endothelial cells in different tissues are quite different. And this makes them functionally different. And actually, this is also consistent with previous funding by Combinate Group, who published the mouse endothelial cell atlas. Interestingly, we also find the tissue-specific endothelial cells show quite distinct biomarkers with that finding in mouse. So this indicates that maybe further functional validation, investigation of the function of those endothelial cells across species will be quite interesting. And actually, the cross-species comparison of the homologous tissue cell types across different species is quite important question. As we know, the brain of non-human primate is an organ that's quite close to human brain. But what's the difference? What's the similarity and difference of these the neuron or non-neuron cell types between human, monkey, and mouse? A very interesting question. So to answer this, we conducted the gene variation analysis across these different species. And interestingly, we found that the non-neuron cell types show quite high gene variations comparing to neuron cell types. And this indicates that maybe the non-neuron type is less conservative than neuron cell types. And this is consistent with previous finding by Allen Institute, which compared the human and the mouse cortex single cell transcription data. And next, I would like to share an example of how can we use the monkey cell atlas to really analyze the disease pathology. As we all know, the COVID-19 pandemic is the biggest event probably in this year. And the cause of COVID-19 disease is the infection of SARS-CoV-2 virus. And the previous study have identified ACE2 as the re-entry receptor of SARS-CoV-2. And this phenomenon can be primed by another serum protease TMPRSS2. The core expression of ACE2 and the TMPRSS2 has higher cells has higher risk of a SARS-CoV-2 infection. So what's the expression pattern of these genes across tissues? So we next performed an analysis for these genes across all tissues we have. And we found that actually first, as expected, we found the ACE2 expression can be detected in lung tissues. For example, the respiratory ciliated cells and alveolar epithelial type 2 cells. Also, interesting, we found the high expression of ACE2 in subcell types of kidney, such as proximal tubular cells and the distal convoluted tubular cells. In contrast, the TMPRSS2 has broader expression across tissues, but we can also see quite high expression of this gene in subcell types of kidney, such as principal cells and the connected tubular cells. Of course, it can also express in subcell types of lung. And we also determined the cell types which co-express ACE2 and the TMPRSS2 expression. And you can see that we can see quite high co-expression of these two genes in subcell types of kidney, such as proximal tubular cells and distal convoluted tubular cells. And well, next we also investigated the similarities and difference of ACE2 and the TMPRSS2 between the human and monkey. Actually, for TMPRSS2, we can see quite consistent pattern of this gene across different tissues, across different cell types between human and monkey. But for ACE2, we observed a quite distinct expression pattern across those cell types. And for example, we can see quite high expression of ACE2 in monkey in ciliated cells comparing to human. And we can see quite high ACE2 expression of in pulmonary alveolar type 2 cells in the lung of human. And in kidney, we can see the highest expression of ACE2 in proximal tubular cells, but with much higher expression of ACE2 in the endothelial cells when comparing to human. And these findings may suggest that the infection of SARS-CoV-2 in these cell types may have different effect. So we next investigated if epigenetic mechanism underlying the, the heterogeneous expression of ACE2 across tissues. So we then focus on kidney, which because it has much higher proportion of cells co-expression ACE2 and TMPRSS2. And we employed the DNB Lab C4 system to generate chromatin accessibility data for kidney, and the clustering result shows that the single-cell ataxic clusters is quite consistent with single-cell asic. For example, we can see large proportion of thick ascending lymph cells and proximal tubular cells in kidney. And we can also detect the rare cell types such as podocyte, stroma cells. And we next check the open region for ACE2 locals across all the cell types. And you can find that we can see the very clear peak, open chromatin peak in the ACE2 locals, but we shrunk the signal in proximal tubular S3 cells, but with less extent signal in proximal tubular S1 cells. And this pattern is quite consistent with the ACG expression across the sub-cell types across in all subtypes of kidney. And we next investigated the transcription factors that potentially binds to the promoter of ACE2. And interestingly, we identified high enrichment of transcription factors such as IRF1 and STAT1 and STAT3 motifs enrichment. And actually, this is quite interesting because that the downstream of cell protective cytokines such as IL-5, IL-6, EGF, or interferon. So probably these are acting on these cell types. And we next investigate IL-6 because the clinical trials have used the IL-6 antibody for the treatment of some, severe COVID-19 patient. So IL-6 is a potent regulator of immune response and is highly expressed in some cell types such as fibroblast, endothelial cells, and tissue macrophage. Interesting, we found quite consistent expression pattern of IL-6R receptor and ACE2. And this can also be reproduced in human kidney single-cell RNA data. And this may indicate that the ACE2 and IL-6 may have a positive feedback loop. So we proposed a molecular mechanism model for SARS-CoV-2 pathogenesis. After SARS-CoV-2 infection contacted to the cells, the IL-6 receptor can activate the transcription factors such as STAT transcription factors, and this further activate the downstream receptor ACE2, and this can have a positive feedback of SARS-CoV-2 infection into the cells. So this is the end of this talk. I would like to thank our collaborators from South China Great Bay Area Single Cell Consortium, and I would like to give my thanks to our colleague from BGI and MGI for the technical support, and also thank people from Professor Miguel Esteban's team for the data analysis and the integration. And thank you all, and I'm happy to take questions.
Thanks so much, Dr. Liu. Excellent talk and very impressive work. We have a first question from a panelist, and everybody, if you have questions, please use the Q&A function to send us your questions. So the first question we have is as follows. If human and monkeys are so similar regarding ACE2 and TMPRSS2
Yep.
And other gene patterns, how come they are so different in their susceptibility to COVID-19?
Yeah, so because ACE2 is a receptor for the SARS-CoV-2, so actually we can see quite high heterogeneity of ACE2 expression in different tissues. And And as you can see in my data, we can see quite high expression of ACE2 in lung and in kidney. And we also see the expression of ACE2 in some other organs such as liver or pancreas with with low expression, but we can still see a proportion of cell types there. And actually, we can see this is quite consistent with some clinical observations. For example, people found that altered kidney function happens in many, COVID-19 patients. And we can also see some patient has, liver dysfunction. So I think this is a quite consistent findings between our data and clinical observation.
Very good. And then I have a question also while we're waiting for other questions here. So I was very impressed to learn more about the macaca atlas. So 40 organs is quite a feast. I'm very impressed that you were able to do that. My question is, were you able to do also spatially explicit analysis, or is this mostly bulk?
Yeah. Actually, we are also just as Jay mentioned this morning. Actually, in BGI, we're also developing spatial technologies based on our because we are developing sequencers as well, or using our sequencer chips, we can really develop high definition spatial transcriptomics. So we have now got quite good results from the technology development, and we have already started the mapping the spatial atlas for some organs of macaque. For example, the brain, we already got the data from the macaque brain, and the resolution is really high. But I hope we can publish this as soon as possible so that we can share the data with this community.
Yeah, this is good. Yeah. And then what set of macaque individuals did you use? How did you pick their age, their sex, how many in total? Could you tell us more about how you selected the population?
Actually, for current data set, we select actually both male and female. We don't have we haven't selected many macaque because the sequence cost is really high, but we already covered replicates from male and female, and we select the adult macaque. But in the future, we are also interested in the sequencing of the features of macaque, and we are also conducting, initiated the developmental atlas of macaque. We are trying to map the atlas for the organ development from the early embryo into the adult organ. Yeah.
Very good. Yeah, I will definitely check out that data. And I was interested to also hear that you reproduced what was found for the mouse, that the endothelial cells really are quite different across the different organ systems. We have another question from Nancy here. She says, thanks for the great talk. Can I assume that if a patient with co-expression of ACE2 and TMPR SS2 will have severe clinical outcomes? So you might also be able to see this in the Q&A panel.
Sorry, let me check this question again.
So Nancy at 11:21. So if a patient has a co-expression of ACE2 and TMPR S2, will they have severe clinical outcomes?
Yeah, I think this largely depends on the, because the co-expression of ACE2 and TMPR SS2 expression cells in different organs are quite different. In some of the organs, we can see a quite small proportion of cells expression this, but I think this is should be consistent in different individuals from my perspective, I think, but we can see a severe or mild case for COVID-19 in the from the clinical observation. But I think probably this is due to the age or distinct the immune response from different individuals. But probably it's not because of the ACE2 and the TMPI ACE2 expression, I think.
Okay.
Yeah.
Thank you. And then Chang Shao Hon is asking, how can you be sure that the differences you observe in endothelial cells in different tissues, which we just discussed, are not due to batch effects or variations between tissue preps?
I think for the batch effect, yeah, this is a very good question. But I think it's not from batch effect because first, all the data are from the same technology platform, and we are actually we have a data generation center, so everything are in the pipeline. So we don't really have big. I don't think we have big batch effect. Also, because we you can see that. We clustered all single cells, and we can see quite only some common cell types show tissue heterogeneity, and this is consistent with previous finding. So, and for the analysis, we use batch effect algorithm to adjust this. So I think the data should be quite convincing. The conclusion should be quite convincing. Yeah.
Is the data published? Can we check
available?
it out? Is this data available?
Yeah, actually we are, I think a couple of months ago, I think in this April, we released the data of 9 organs already. And the people can check our data from the website of, I think, the Broad Institute. We also submitted the macaque data for 9 organs. And we also have this data available in China National Gene Bank. We also have a single cell portal so that people can check their interested genes. And I think we are still preparing the manuscript. So I think probably we will release the data quite soon in